So I'm going to present you the results from my master thesis realized under the direction of Vincent Moore and Menez-Dregan I, and with the precious help of David Collonges. A technomorphometric approach of cleavers has been tested as part of this work through the study of two series from different chronological and geographical contexts, the cleavers from Menez-Dregan and Lanne-Darré sites. The important variability of Middle Pleistocene lithic industries is now accepted, but is still very complex to grasp. The production of large flakes, used as blank of some characteristic macrotools, takes part in this diversity. The study of cleavers could, in particular, help to a better characterization of this technocultural landscape. So the definition of cleaver is still under debate, but we choose to use TIC-1, which can clearly identify a technical phenomenon. According to this determination, a cleaver is always produced from a flake and shows a non-retouched transversal cutting edge. This involves an anticipation of the technical moves, a mental representation of the applied method, and therefore the mastering of a specific conceptual scheme of production. So in Europe, cleavers are mainly represented within the Iberian-Acheulean industries in the southwest of the continent, to which is related Lanne-Darré site. In contrast, the north and east of the continent seem largely devoid of cleavers, except for some sites like Menez-Dregan in the Armorican Massif. A technological analysis of the rare cleavers from Menez-Dregan highlighted some technical and petrographic similarities between these locally atypical pieces and cleavers from the southwest of the continent. But technological observations only allow no interpretation about possible technocultural links. In fact, other factors might also have influenced those productions, such as raw material availability. To assess the technocultural implication of cleaver, this tool must be considered according to several interpretative frameworks, and its place within the mental templates of human groups needs to be clarified. Morpho-functional analysis aims precisely to identify functional goals behind lithic productions by the characterization of technical, morphological, and morphometrical convergences. Regarding the cleaver, something to explore is a technomorphometric analysis of its transversal cutting edge, directly resulting from the debitage of the blank. However, cleavers show other ridges on their circumference, which can also be seen as possible transformative areas. So this study aims to bring new considerations about the technocultural places of Menez-Dregan and Lanne-Darré's cleavers, but its principal objective is the establishment of a technomorphometric approach for cleavers, which could also be used for the study of all macro tools from this kind of industries, integrating the search for common or specific cultural features within the European Middle Pleistocene technical systems. Menez-Dregan has been excavated for about 30 years until 2021. More than 154,000 lithic artifacts highlight lower Paleolithic occupations, with a transition between lower and middle Paleolithic in the upper levels. Within 12 macro tools with transversal cutting edge, only 5 cleavers were identified in our study, in non-successive levels dated between MIS 10 and 8. The study also includes a sample of 47 cleavers from Lanne-Darré, more abundant and diversified than the sample from Menez-Dregan, to support a common methodological issue in a complementary way. Lanne-Darré is an open-air site located in the French Pyrenees and Piedmont, excavated between 1997 and 2003. More than 3,000 lithic remains, which testify of one occupation, were collected in 2 archaeological levels in secondary position. Cleavers from Lanne-Darré are mostly made from local quartzite pebbles, and those from Menez-Dregan are all from littoral pebbles. Several layers of analysis are experimented here. So first, the observed entities are cleavers in their entirety. Petrographic and technologic observations allow to set groups of cleavers whose morphometric variability can be analyzed. Cleavers technotypes from Jactixis typology in particular are determined, analyzing the transversal cutting edges production. Here we can find type 0 with a non-prepared transversal cutting edge before the debitage of the blank, types 1 and 2 with a prepared cutting edge by one or several removals prior to the debitage, and type 6, which means cleavers made on Combewa flakes. 3D scans are also used to take and process morphometric data. HGMT 3D software allows to extract some metric measurements and to apply geometric morphometric analysis. The variability of cleaver's shape, excluding size and orientation criteria, is observed with the use of a generalized procrust analysis followed by a principal component analysis. The study then focused on the ridges, the observation of production modes of the entire piece's circumference allows to identify technical units which can be analyzed independently according to morphological and morphometrical criteria. Artifact 3D software is used to measure the mean edge angles of those technical units, for example, using the faces areas geometries. For both scales of analysis, statistical tests are used to check normality of the distributions and to confirm if links exist or not between the samples. So on the first scale analysis, only Lanne-Darré cleavers are numerous enough to allow reliable statistical comparisons. Some criteria such as blank debitage direction or technotype do not seem to impact Lanne-Darré cleavers measurements, but others such as quarter type or blank type sometimes show a significant link with those measurements. On the other hand, the principal component analysis reveals low discriminating morphological criteria, and no significant trend in these shape variations seems to appear from petrographic or technical groups of cleavers. On the second scale analysis, when we compare the various ridges technotypes within each sample, similar links are perceptible between production modes and morphology or morphometry. For example, mean angles of ridges directly coming from the debitage of the blank are always globally more acute than mean angles of ridges coming from prior removals or from a retouch stage after the debitage. Then we distinguished 5 edges morphotypes that appear recurrently, depending, for example, of the wire regularity and angle. These morphotypes are consequences of technical movements performed during different stages of cleaver production, and the combinations of edges morphotypes and technotypes allowed to identify possible techno-functional units. Then only recurrences observed through link organizations of these units on the cleavers allow to consider specific functional goals. Figures from Menez-Dregan show a common technomorphofunctional structure, but the anecdotal nature of this sample and the low technical constraints required to produce this kind of cleavers do not permit to reject with certitude the impact of raw material availability on technical choices. In contrast, 4 technomorphofunctional groups are highlighted in the Lanne-Darré sample. So we have cleavers with flattened base, others that can be defined by an overall angular circumference, others with lateral back, and others with 2 transversal cutting edges. The important technomorphometric uniformity of the transversal cutting edges on these cleavers could reflect intent use specific to this part of the tool, but the diversity of the organizations observed on the other parts of the pieces allows to consider several functional goals within a shared production concept suitable for cleavers. Questions about the relevance of the tested approach when looking for cultural traditions have been raised after all. This study shows, for example, that the entire shape of cleavers strongly related to the blank's shape itself can't reflect clearly functional goals of production. A smaller observation level is therefore essential, but it is also challenged by a lot of difficulties. Another fact highlighted is that neither size or shape of cleavers or the technomorphofunctional groups identified here show relations with Jack Tixier's technotypes. On one hand, Tixier's classification seems partly incomplete, only based on the modes of production of the transversal cutting edge, and on the other hand, even if it is clear that technomorphometric analysis allow to grasp the functional potential of cleavers efficiently, the criteria defined to characterize the technomorphofunctional variability of this tool need to be refined. To conclude, it is important to keep in mind that the cultural implication of this kind of tool cannot be reached only through the possible intent to use that it reflects. The available raw materials influences in particular must be precised. In addition, all the other parts of the industries must be observed as well to understand the place of cleavers within technical systems. The technomorphometric approach that we tested here could precisely allow to analyze all the associated toolkits toward a better comprehension of the status of the characteristic macro tools represented within Middle Pleistocene industries. Thank you. Don't hesitate if you have any questions.
Thank you so much. So yeah, I'm Abigail PARKER. I'm just finishing my PhD at the University of Cambridge. And today I'm going to talk to you about my work studying reptile body size in the Turkana Basin of Ethiopia and Kenya. I initially became interested in reptile body size because it is often taken as an indicator of tropical paleo environments. My aim was to rigorously test the relationship between reptile size and paleoclimate, treating it as an ecometric trait, which is a trait whose distribution in a community is correlated to habitat factors across space and time. Reptile size is a strong candidate for an ecometric trait because reptiles, as ectotherms, rely on their body surface area for thermoregulation. I began by measuring reptile fossils from these dated sites on the map here across Ethiopia and Kenya. And these sites are extremely well sampled because there have been hominid fossils found there, so a lot of field work has been done. And the reptiles that are found in this region are shown in these photos. So we have a diverse group of crocodilians, like in these skulls, turtles, for example, this soft-shelled trionicid shell, as well as snakes and lizards, specifically large varanid lizards. And these two vertebrae at the bottom are a python and a varanid that I found in the field. So most of the work that I did with these specimens was developing total body length and body mass estimates from partial fossil material. On the left, for crocodilians, here are some relatively complete skulls. So the top two are variably assigned to the genus Crocodylus, which I refer to as the Crocodylus morphotype here. And below, we have the extremely long-snouted taxon Euthecodon. There are reliable regressions to estimate total body length from skull length for these crocodilians, but when I had fragmentary or postcranial material, like these specimens on the left here, I had to develop regressions based on more complete material. Here I have a bit of detail on one of these regressions. There were a lot of specimens that preserved just chunks of the maxilla or dentary of that very long snout of Euthecodon. So I developed a regression estimating total skull length based on the spacing between tooth alveoli. This spacing remains relatively constant along the rostrum, as plotted on the right here. I excluded the identifiable two most anterior alveoli, which are wider spaced, and related the average tooth spacing from complete museum specimens to their total skull length. This regression enabled me to reconstruct body length and mass for many Euthecodon specimens, like the one on the top left here. Some turtle specimens had the entire carapace preserved, which is a great measure of body length, like the soft-shelled trionicid that here in the Shangura formation is weathering out in that photo, or this giant tortoise, which is preserved on its back, so the limb and girdle bones are exposed. But there were also many partial turtle specimens, like the ones on the right here. So I developed regressions to estimate total carapace length from limb bone lengths, articular surface widths, plastron lengths, and widths of trionicid costal carapace elements. Once I had these body size estimates for all my specimens, I began testing for trends in maximum size change over time in the Turkana Basin. Here, I've plotted the maximum body sizes of each crocodilian morphotype and turtle taxon at dated sites starting from the Miocene across the Turkana Basin. In the top graph, we can see the crocodilian size has remained relatively constant, except for several very large specimens of Euthecodon in the Pliocene and Pleistocene. This taxon, as well as several species in the Crocodylus morphotype, went extinct prior to the Holocene, when Crocodylus niloticus became the only species alive in the region, and its maximum body length is only around 4 meters. For turtles, the variation in size between sites is largely driven by whether terrestrial tortoise fossils are sampled. From the Miocene to the Middle Pleistocene, tortoises over 1 meter in carapace length were present, while the modern sulcata tortoise that lives in the region doesn't exceed around 85 centimeters. Both Euthecodon and the giant tortoises here are the largest reptiles known globally from these time periods. So the drop-off in reptile maximum size that occurs going into the Holocene here represents a reorganization of faunas. Next, I focused in on the Shangora Formation, where I did field work for 2 years studying these reptiles with the OMO Group research expedition. This formation has been really intensely sampled due to interest in hominid fossils that it has yielded. So the composition of its mammal faunas and proxy records for vegetation change are well known relative to many other formations. My aim was to use this available data as a case study to understand the environmental context for change in reptile size between members of the formation. I found that sampling counts for reptile fossils were not significantly correlated to maximum size across the formation, which allowed me to rule out sampling as a driver of size change in the reptile record. Here, we can see that for terrestrial turtles in the green line, size actually drops when sampling for turtles is the highest. The sizes of Euthecodon, which is the dark orange here, and Crocodylus in the light orange, change in a very coordinated fashion across the formation, though Euthecodon is always larger than Crocodylus. For lizards and snakes, sampling is very poor. There's only a couple specimens, so I don't analyze trends in these groups any further, but you can see that in the early Pleistocene, pythons do reach very large size in the Shangora. I conducted statistical tests for correlation between each of the reptile group's size and between each group's size and paleoenvironmental indicators, including reconstructed lake levels, carbonate isotopes, and mammal faunal composition. In most cases, there were insignificant correlations, with the exception of a strong negative relationship between the size of terrestrial turtles and pythons, and a strong positive relationship between the size of aquatic turtles and lake level in the region. Here, we can see how aquatic turtle size drops and then rises again as the lake level does the same. This is based on paleo lake level volume estimates for Lake Turkana from Nutz et al. 2020. And it makes sense that aquatic turtles grow larger when there's a greater volume of aquatic habitat available to them. For terrestrial tortoises, shown in the green line here, we see again there's that drop in size in members E and F, and I believe that this may be due to changes in vegetation. So this red and purple line are delta C13 values from carbon isotopes in Shangora paleosols and also from bovid tooth fossils, which are the most abundant group of herbivore mammals. So higher values of this isotopic ratio indicate a higher proportion of C4 plants, which are typically associated with more open habitats. So over time, the Turkana Basin is trending towards more open grasslands. But specifically from around 2.5 to 2.2 million years ago, this trend was temporarily reversed. With these more closed C3 plant-dominated forest, tortoise size decreases. This makes sense because the largest modern tortoises inhabit open grasslands. They may have historically relied on such grasslands for their grazing diet or been challenged for locomotion in more closed forests. Also, the negative correlation that we saw between tortoise size and python size may show that the increased size of the snakes as tortoise size drops is because there's a greater availability of habitat for pythons in more closed forests. Much like the aquatic turtles, crocodilian size increase may occur as lake level rises, which would make sense because the lake could support more prey resources for crocodilians when there's a greater lake volume. Here we see that the greatest increase in size for both crocodile groups is in member F as the lake level is rising. However, this large size is not maintained through the lake high span that occurs in member G, and this demands some further explanation. Overall, comparison with these paleoenvironmental records shows that available resources in a taxon's favorite habitat enable increases in maximum body size. So next, I developed a range of models relating turtle body size specifically to temperature and precipitation to estimate these paleoclimate values from my measurements of turtle fossils. Turtle and other reptile size is hypothesized to be greater in warmer habitats, This is because reptiles have a low metabolic rate, and in all groups, metabolism per unit mass decreases at larger body sizes. Low body temperatures in ectotherms also lead to lower metabolic rates. Therefore, large-bodied reptiles in cold climates would have very low metabolic rates. And past work had predicted that processing energy that slowly would not allow them to survive. To maintain high enough metabolic rates, large reptiles are hypothesized to require warmer habitats. So this pattern is confirmed when I collected data from modern turtle communities. These are globally sampled communities, and we can see that average size increases with temperature for both the aquatic turtles, which are on the left, and terrestrial tortoises, which are on the right, although there's really a lot of scatter in this relationship. So this linear model of turtle size versus temperature is one of two methods that I used to predict temperature based on turtle size. The other method is maximum likelihood modeling, where modern training communities are binned by statistics describing their turtle body size distributions. The most likely temperature at those sampling points becomes the predicted temperature for a community that has a similar size distribution. I compared the accuracy of these models at modern test points and found that the maximum likelihood model predictions better matched the observed temperature and precipitation values than the predictions from the linear models. Although both of these models have a pretty large error, which is a couple of degrees Celsius or around 50 centimeters of mean annual precipitation. Because the maximum likelihood models performed better in the modern test points, I used these to estimate paleotemperature, which is in red here, and paleoprecipitation, which is shown in purple, for each member of the Shingora Formation based on the size of the turtles, which are also plotted at the bottom of this graph. So I plotted predictions based on models that are trained across global sampling points, as well as models trained only in modern African turtle communities. And their predictions are plotted here alongside some independent proxy estimates, which are shown in the dotted lines from other authors. And these temperature estimates are several degrees lower than the modern temperature in the Turkana Basin, which may have been the case in the Pleistocene, though some of the values, which are around under 18 degrees Celsius, are unlikely to have occurred in this equatorial region. The precipitation estimates are in the neighborhood of other reconstructions, with values ranging from around those of the highly arid modern Turkana region to much higher precipitation in some of the members. And the timing of these really high precipitation estimates matches periods that have been reconstructed as more humid by other authors based on mammal faunas and geologic sampling. So to summarize, I don't view these ecometric models as highly accurate to reconstruct these paleoclimate variables, but they can provide estimates for fossil sites where you have sampling of turtle fossils. And as this shows, they can identify where large-scale changes in temperature or precipitation occur. So just to conclude, my study of the reptiles of the Shingora suggests that reptile body size increases when preferred habitats of taxa are widespread. While ecometric models have some utility for estimating paleoenvironments, the direct effect of climate variables on this reptile trait is likely less than the effects that are mediated by ecosystem-level changes in resource availability. I really want to thank all of the collaborators that I've worked with on this project, especially everyone that I worked with in the field, in the OMO, and in West Turkana. And thank you all so much for listening. I'm happy to answer any questions.
To present you a communication on Plio-Pleistocene canids and the contributions of an ecomorphological study to the restitution of their place in the food web. The Guild of Extant African Canids includes 4 genera and 5 species. The smallest representatives are the genus Vulpes with the Cape fox and the genus Otocion with the bat-eared fox. Medium-sized canids are represented by the genus Lupulella, which has 2 species, the black-backed jackal and the side-striped jackal. Finally, large canids are represented by the genus Lycaon. Fossil canids in southern Africa are exclusively derived from the cradle of humankind sits in the South Africa, which is known to hold an extremely rich paleontological record, including the oldest and best preserved Australopithecine remains, such as Littlefoot at Sterkfontein. The fauna associated with these remains is therefore essential to the characterization of the paleoecosystems in which these hominins evolved. Carnivores, in particular, due to their position as predators or in direct interactions with them. Characterizing their ecomorphology and their role in food webs will help to better understand the behavioral evolution of hominins. The Plio-Pleistocene carnivore guilds of Southern Africa include a higher species diversity than the present-day guilds. This observation is particularly true for canids. 7 genera and 14 species are identified. Small-sized species are represented by the genus Vulpes, Otocion, and Prototocion, the foxes and the bat-eared foxes. The numerous medium-sized canids are represented by the 2 species of jackals currently found in Southern Africa. A chronoform has also been identified for the black-backed jackal. 2 species of the genus Canis whose specific value has been debated because of their occasional presence on the seats are mentioned. These are Canis brevirostris and Canis anticus. A last genus is present for this syringe. It's Nycterotes, the raccoon dog, which today is an exclusively Eurasian species. A canis of intermediate size to the jackal and large fossil canids, similar in size to the current lycaon, has recently been identified. It's Canis ewiti. Finally, large canids are represented by the fossil wild dog and a large dog, Canis atrox. The important specific diversity of Plio-Pleistocene canids, especially for those of medium size, leads to discuss the validity of certain forms. We can then wonder whether if this diversity is a reflection of past biodiversity or a reflection of wide intraspecific variability. This question seems justified since a significant morphological variability is observed in the extant register for the black-backed jackal. The general shape of the first upper molars is extremely variable, especially at the level of the construction of the heel, which is more or less marked, depending on the individuals. This variability is also found in the fossil record. Moreover, a metric analysis shows no difference in size distribution between the black-backed jackal and the subspecies, because the 2 shapes overlap completely. It's the same for Canis brevirostris. Further study of the morphology of this fossil species is needed to allow reassessment of specific and subspecific attribution. This generic and specific diversity covers as many specializations and behaviors. Large-bodied species, such as wild dogs, show an adaptation to hypercarnivory with a carnivorous diet. And as body mass decreases, the species are more omnivorous with a diet that includes, alongside small prey, berries, fruit, and insects. The morphofunctional study of a new species identified on the site of Cromdry Bay Member 2 led to the identification of a canis with mixed characters, further complicating the trophic chain. It's a dog of intermediate size with both typical morphological traits of wild dog-like, with second characters, such as the small additional cusps on the lower P4, and both adaptations to grinding, with a bicuspid talonid for the lower carnassial. We can arrive at a fairly detailed vision of the place of the canids of the cradle within the trophic chain, because all of these ecomorphological studies relate to fossils from this area. However, it covers an area which represents only 0.05% of the total area of South Africa. It therefore seems difficult to consider the cradle as representative of the paleoecosystemic context of Southern Africa as a whole. This is why research programs are underway for other karst areas in Southern Africa, including the Guyaba Hills sector in Botswana. The remains of Botswanan canids were cast and subjected to morphometric analysis with an ecomorphological approach. Significant differences are noted with their South African contemporaneous counterparts. Concerning the jackals represented here by the two casts, they are morphologically similar to the blackback jackals and differ from the side strip jackal. This morphological observation is confirmed by the metric, which shows that the two specimens from Guyaba are in the high values of the blackback jackals and do not cover the values of the side strip jackals. However, when we are interested in a functional length, here the crushing part of the maxilla, we realize that the jackals of Guyaba integrate this time more with the values of the side strip jackal. The latter has the crusher part of the teeth more developed than the blackback jackal because it has a more general diet, while the black jackal tends to be more omnivorous. So the blackback jackal from Guyaba shows an ecomorphological particularity, which will be a more marked adaptation to omnivory with a more developed chewing part. Fossil raccoon dogs are larger than the extant one. The Guyaba raccoon dog differs from the extant species by ancestral characters called plesiomorphic. The M1 has a trigone with cusps of subequal size, whereas in the extant one is bigger than the other, and the protocone of the P4 is much smaller than for the extant one. Nevertheless, its size is significantly smaller than that of the fossil species, since it fits perfectly into the field of variation of the extant species. So the Guyaba raccoon dog is a fossil form, but whose size is significantly smaller. All of those ecomorphological differences observed with the morphometric study are confirmed by the 2D geometric morphometry. Semi-landmarks were placed on the contour of the M1s in order to study the conformation of the heel, crushing part of the tooth, compared to that of the trigone, cutting part. The first 7 variables from the PCA were then used to perform a discriminant analysis. This statistical test makes it possible to predict membership in predefined groups from a series of variables. The 5 groups obtained are Lycaon, Icterotes, Canis, Lupulella, and Vulpes. The discriminant analysis confirms the morphometric observation made on the Guyaba individuals, since the 2 Botswanan jackals fall between the Canis and Lupulella group, while the raccoon dog almost fits into the Canis pool, thus showing a significant difference in their conformation, while by evacuating the bias of a deformation induced by a difference in size. In view of these preliminary results, it now seems necessary to identify environmental or ecosystemic variables in order to explain these morphological variations produced on the dental material. A correspondent analysis was performed to assess similarities and differences in carnivore gill structure between the cradle and Guyaba seats. Guyaba approximates the seats that are in the top left square, which are associated with the variables Canis and Lupulella, so the jackals. However, the Guyaba seat is the only one to present a big-sized dog, jackal-raccoon dog as co-occurrence associations, which makes it possible to infer a structural difference with the carnivore guild of the cradle seats. The difference is the presence of Canis ewiti, which would occupy an ecological niche comprising a more carnivorous diet, thus triggering a trophic cascade system. It occupies the place of a mesocarnivore with a carnivorous tendency, thus allowing jackals a more developed adaptation to omnivory, which results in a development of the grinding part of the teeth, what could have led to an adaptation of the raccoon dog size with a smaller body size and an even more opportunistic place with an omnivorous diet with vegetarian tendency. In conclusion, the case of canids illustrates the need to infer an ecological status to species in order to identify specific ecological niches, especially since potential remains of hominin will be present in the area. The paleoanthropological study is still in progress, and we need confirmation. But Moreover, it's the characterization of food webs that will allow the recognition of the paleoecological status of hominins. So thank you for your attention.